Biomaterials are any engineered substances that are designed to interact within a biological system to direct medical treatment. The major difficulty in biomaterial research is finding material that will both address the medical treatment as well as not cause detrimental damage to the host. Scientists have tested the applicability of biomaterials made from metals, ceramics, glasses, and polymers. The use of polymers is particularly enticing because polymers provide a wide range of physical properties which can make them adaptable to many applications. Some of the most popular usages for polymers in the biomedical realm are for electronic devices, tissue engineering, and drug delivery. Biomedical electronic devices operate by transmitting a chemical signal into an electrical current. Conducting polymers, or CPs, have the unique ability to conduct electricity, while still maintaining the advantageous processing and mechanical characteristics of a polymer. One of the most common uses of CPs is in the field of biosensors. Biosensors, such as a blood glucose monitor, are made up of a base polymer which contains a sensing element and a transducer. The sensing element, such as an enzyme, serves as a receptor for analytes, such as antigens, and produces a chemical signal. This signal is received by the transmitter, which in turn transforms the chemical signal into an electric signal. This signal is then translated to a quantifiable output, such as glucose concentration. Polymers are also used in tissue engineering. Tissue engineering uses biomaterials that interface with biological systems to help cure, change, or replace failing organ tissue. Tissue engineering begins by creating a 3D plastic mold called a scaffold or matrix. Every part in the human body has unique function. Thus, any artificial body part will require a scaffold with proper structural and chemical and mechanical needs. This figure shows some polymers and their applicability to the human body. Once the scaffold has been created, stem cells and nutrients are spread across the scaffold. Tissue growth ensues, and the final product is a body part ready for implantation. Aside from electronic devices and tissue growth, polymers are also used in drug delivery. Polymers are used to control the location and rate of drug delivery. This is called controlled release. When delivering a therapeutic agent to a location in the body, we don't want the compound to be broken down before it reaches its destination, but we also don't want it to stay in the body forever without being used. Controlled release is the use of a biodegradable polymer to encapsulate or tether a drug and degrade at an optimum rate so that the body can make full use of the drug. Controlled release is done using polymers with a hydrophilic or water-loving end and a hydrophobic or water-hating end. When placed in water, the hydrophobic regions want to hide from the water, and many times they form what is called a micelle, which is basically a tiny sphere with hydrophilic ends facing the water and the hydrophobic ends hiding inside. In drug delivery, this can be used to encapsulate a drug within the sphere, and as the hydrophilic shell degrades over time through controlled release, giving it time to get to its destination, the drug is slowly released from the micelles. Another means of drug delivery is a matrix-based system. Here, a non-degradable polymer patch, implant, or injectable hydrogel is used to distribute drugs into the body. Drugs are placed inside of the polymer matrix, and the drugs will slowly diffuse out into the patient. However, sometimes this is problematic since surgery may be required to replace a depleted polymer. To reduce the frequency of surgery, researchers are developing degradable matrices with drugs tethered to the polymer. In this system, the drug will be delivered as the polymer dissolves. As you can see, polymers play a crucial role in biomedicine and will continue to be heavily researched in the future.